This follows from the step 1 which we covered in the previous video. I highly recommend you watch it before this one. So step 2, or the first step of initiation, is the formation of pre-initiation complex, which then leads to state of the initiation of transcription. And this finally causes RNA polymerase to escape and clear the promoter. But it pauses before proceeding to the step of elongation. So we will see how these steps make up the initiation stage of the transcription in eukaryotes. So let's start with pre-initiation complex formation, which also happens to be a rate-limiting step in the eukaryotic transcription. Just to set up some background, we have the DNA which is wrapped around histones, and these histones are displaced at the promoter region by various mechanisms that we discussed in the last video. And once the promoter is accessible, the GTFs and the RNA polymerase can assemble at the promoter, which starts the initiation stage. So, pre-initiation complex is nothing but a sequential assembly mechanism through which the general transcription factors and the RNA polymerase are recruited at the promoter region. Also, just to keep things simple, we'll only talk about RNA polymerase II associated initiation, because RNA polymerase II happens to be the most studied RNA polymerase. Alright, so once the histones from the promoter regions are displaced, and the promoter region becomes accessible, the first GTF to approach the promoter is TF2D, which is a multi-subunit factor whose main components are TATA binding protein, TBP for short, and TAFs, which are TBP-associated factors. And there are multiple TAFs that can associate with TBP. You may recall from the promoter structure video that TBP binds the TATA box. So just to sketch out the promoters that we will focus in initiation, we have the BRE elements, and sandwiched in between is the TATA box. And at the downstream end, we have the initiator. Again, you should check out the promoter structure video. I have linked that in the description if you want to know more about these promoters. So TBP, the TATA binding protein, can either bind the TATA box alone, or it can come with the TAFs in a bigger complex and then bind to the TATA box. The TBP-associated factors, on the other hand, are often contacting the initiator motif. So this TF2D is really just binding the AT-rich promoter sites, which are the TATA box and the initiator. Now what is interesting to note here is that the TBP actually searches for AT-rich sequences along the DNA, but it only settles when it finds the TATA sequence. So why does it settle for TATA sequence? Well, the way TBP works is by locally distorting the DNA in the minor groove. And because adenine and thymine bonds are easier to distort, it settles for this TATA region. And in the process of this local distortion of DNA, the TATA binding protein creates almost like an 80-degree bend in the DNA. And this local distortion is done by these two hooks or edges which are present in the TBP. And these consist of phenylalanine amino acid residues. And phenylalanine is an aromatic amino acid which can easily intercalate between the DNA. So what we have concluded is that TBP causes a structural change at the TATA box, in particular an 80-degree bend. And this conformation of DNA bend is not super stable. And therefore, to help this, we have another GTF factor that comes in, which is the TF2A. Just a side note that TF2A, or transcription factor 2A, is not a strict GTF, meaning that transcription can occur even in its absence without much difficulty. So TF2A binds TBP and stabilizes the TBP TATA box complex. So just to complete the picture, TF2A is simply a scaffold GTF that locks the TF2D in place at the promoter region, so that the bending of the DNA, the distortion that we talked about, can be more stable. Another thing that happens as soon as the bend is created is the recruitment of TF2B. TF2B, as you may recall from the promoter video, binds at the BRE elements, and it binds in the major groove of the DNA. TBP perhaps is one of the rare proteins that bind the DNA in the minor groove. All other DNA binding proteins search for major grooves, because these are the grooves which have more space in them. Anyways, I want to stress here that TF2B binding is independent of TF2A, so it happens as soon as the bend is created by the TBP. Many textbooks will show that TF2A comes first, but it is an oversimplification of a dynamic process. Let's complete the picture of TF2B binding in our complex. Here, there's one thing to keep in mind, that is the B reader unit of the TF2B complex, which faces the upstream side. So what we're really trying to establish here is that there's an asymmetric binding of TF2B, which is underscored by the specific positioning of the B reader unit. And this reader unit determines the polarity of transcription. That is, whether the transcription should occur in the downstream direction or the upstream direction. Another important feature of the B reader to keep in mind is that when the RNA polymerase is recruited, it blocks the RNA exit channel and a fair amount of its active site. Compared to prokaryotic transcription, if you recall, this function of blocking the active site and the RNA exit channel was largely done by sigma-3,4 linker protein. Again, you should check out the promoter transcription first if you haven't watched that already. The link is in the description. And we'll see TF2B in action when we talk about the step 3, which is the initiation, and step 4, which is the promoter escape and clearance. Now, because we said that TF2B is in a way inhibiting RNA polymerase function by blocking the RNA exit channel and the active site, this suggests that enzymatically TF2 inhibition is a rate-limiting step in the process of transcription initiation, which, as we will see shortly, determines the rate of abortive transcription. So now, when this stable complex of TF2B is formed, the RNA polymerase II is recruited. But the problem, as we have seen from the introduction video, is that it cannot simply bind the DNA, because RNA polymerase has no affinity to the DNA by itself. To aid in this process, TF2F comes in. So you can think of TF2F as an accessory GTF, which has one unit that attaches to the RNA polymerase, and then the remaining surface, which has the affinity towards TF2B at the promoter. So this complex of TF2F and RNA polymerase is what we call a free state complex of RNA polymerase. The paradoxical thing about TF2F is that in its free state, it really acts as an inhibitor to the RNA polymerase binding, preventing it from binding the DNA. But once TF2F binds TF2B and TBP or TF2D in the complex, it can bind there and bring RNA polymerase with itself, so it no longer acts as an inhibitor in that sense. Now, just to bring all of this into a coherent picture, I'll draw this out. We have the TBP and the TAFs bound to the TF2A and TF2B, and then the TF2F sits very close to the TBP and the TF2B complex, with the RNA polymerase facing to the downstream direction. And this positioning is done with the help of B reader unit part of the TF2B, which now, as soon as the RNA polymerase is recruited, the B reader goes into the exit channel of RNA polymerase and it blocks it. And it also blocks some part of the active site of the polymerase. Okay, so once TF2F is loaded onto the complex with the RNA polymerase, the TF2F can attract the next set of GTF, which is TF2E. TF2E actually functions very similar to TF2F, in that it also acts as an accessory factor for TF2H, and it brings TF2H to the pre-initiation complex. So once again, TF2E is simply a loading factor for TF2H. TF2H is the biggest and the most complex general transcription factor of all, and it is a very important factor when it comes to the process of initiation, and we'll see its function very soon. So the point we're making with this assembly is that TF2E, which is a loading factor, binds this large TF2H in the free state and brings it to the complex at the promoter region. Now again, let's just clarify the picture and put TF2H within our complex. Note that TF2E is sitting next to TF2F, and that's because F is what attracts E. And TF2H, which is the major general transcription factor, actually sits downstream of the RNA polymerase, which is right next to the template DNA entry channel. And we'll see why this is important in the next step. Now, the final piece of picture that we're missing is that the assembly of GTFs is usually happening in a three-dimensional conformation, where the long-range interacting enhancers, which is typical of human genome, contain these important transcription factors and complexes such as the mediator proteins, which are always talking to the TBP, TF2H, and even the C-terminal domain of the RNA polymerase. These factors that are bound to enhancers can be pioneer factors or kinases and many other complex proteins that can help in the process of transcription initiation. So this entire complex is known as the pre-initiation complex, and that's the first step of initiation. Sometimes people will not refer to the enhancer proteins when explaining the pre-initiation complex. But that picture where enhancers and mediator is missing is known as a basal pre-initiation complex. Basal because there is no additional help beyond the promoter region. And that might be true in simpler eukaryotes, such as yeast, but a true pre-initiation complex in context of humans, where things are more complex, the pre-initiation complex has enhancer-bound proteins in the picture as well. And now we're ready to move on to the second and third step of initiation, which is the initiation step followed by a promoter escape, clearance, and a proximal pausing. So I'll just quickly sketch out the pre-initiation complex because that is where we're starting from. So you have the GTFs and the enhancer-bound factors that constantly talk and interact with each other. So the initiation step we can define as the transition from pre-initiation complex, which is a closed complex, to an open complex. We call pre-initiation complex as closed because the active site of RNA polymerase is blocked, the template DNA is still base paired, and the RNA polymerase is not making any RNA. But the open complex means that the active site becomes open to activity. So the big question now becomes, how does this transition from closed state to an open state happen? The answer is that it happens due to the activity of TF2H. TF2H, as we just said before, is the most complex GTF of all. And to add details to its structure and functional capacity, it consists of 3 independent subunits. That is a DNA helicase, a DNA translocase, and a kinase. In humans, these individual subunits or proteins are usually referred to as XPD, XPB, and CDK7. If you're familiar with cell cycle enzymes, CDK is a cyclin-dependent kinase that is also involved in cell proliferation activity. But here, we're looking at its function in terms of regulating transcription start. Just a quick side note. In yeast nomenclature, XPD and XPB are known as RAD3 and SSL2. I'm not going to use this yeast nomenclature, but if you come across these names with respect to TF2H, don't get confused. Now, functionally speaking, the helicase works from 5' to 3', and the translocase works from 3' to 5'. Now, to explain the transition from closed to the open state, I will draw out the GTF that are relevant only to the understanding of this concept. So just because I'm not drawing the rest doesn't mean that the other GTFs have disappeared in this process. So at the TATA box, we have our TATA binding protein, which is linked to the RNA polymerase in our pre-initiation complex, with the TF2H sitting near the mouth of the RNA polymerase. And TF2H is in turn tightly attached to the RNA polymerase. So these 3 components form like a chain. The key thing to focus now is that TBP and RNA polymerase are in a tight linkage, almost as if the two are fixed in their spot. TBP is very specific to TATA box, so it'll not let go of the TATA box, but RNA polymerase has no specificity to the DNA, and it is a processive enzyme, so it really wants to move forward, but TBP is not going to let it go. So somehow the RNA polymerase has to move or dissociate away from TBP, and therefore also the promoter region, to start transcription. Now, the idea of RNA polymerase movement is actually very similar to what we have seen before in prokaryotes, which is the process of DNA scrunching. The eukaryotic DNA scrunching is quite a bit different from prokaryotes. So in case of eukaryotic transcription, the TF2H translocates along the DNA, and this causes the DNA to unzip. Well, why does it unzip? Because we've just said that TBP is fixed to RNA polymerase. And this in turn fixes the TF2H to the RNA polymerase. So if TF2H wants to move forward, it really cannot move. Instead, what happens is that the DNA gets pulled inwards because of the movement of TF2H. And because TF2H is sitting near the template entry channel, As a result of the movement, the DNA that is unzipped ends up in the activity center of the RNA polymerase. And this is what we call a DNA bubble formation. So I will stress on this once more because this is crucial to understand. The XBB subunit, which is the translocase part of TF2H, tracks the DNA forward, but it cannot move forward because it is connected to RNA polymerase, which is in turn connected to this fixed TBP at the TARA box. So as a result, the DNA unwinds and it ends up in the activity center of the RNA polymerase. Another point that I want to stress here is that many textbooks will tell you that XBD, which is a helicase, is involved in this process. Or sometimes they will also say that XBB is actually a helicase and not a translocase. But all of that information is outdated. These differences between XBB and XBD are crucial to understand. A translocase creates a torsional force when it tracks the DNA, whereas the helicase breaks bonds by unwinding the DNA. A translocase doesn't really unwind the DNA. Here, XBB may appear like a helicase given that DNA is being unwound, but studies have shown that XBB cannot really unwind DNA if you try to test it. So, back to where we left. We have now created this bubble in this active site of RNA polymerase due to the movement of the translocase. Now I want you to recall that the B reader subunit of TF2B is partially also sitting inside the active site and is also blocking the RNA exit channel. So after the bubble is formed, the RNA polymerase tries to make RNA using this open strand that is now available. But this B reader, which is sitting in the activity site of the RNA polymerase, blocks the RNA polymerase, and therefore it cannot continue with the RNA synthesis. And sometimes the RNA fails and releases these short 3 to 5 or sometimes even 10 base pair long transcripts in this process. And this process of aborting the short RNAs is called the abortive transcription. And this is really due to the blocking activity of TF2B, which prevents the RNA from elongating further. Now, this blocking of B reader is also very specific. It only takes up a partial space in the bubble. It doesn't block the entire unbound template. And this actually ensures that RNA polymerase begins transcription at a very specific position. And that now becomes the +1 site of the RNA. Otherwise, there's no way for RNA polymerase to know where to start. The +1 site determination only happens in context of GTFs. And now that some RNA is initiated and the +1 site is ensured, we call this step as the initiation step, even though there's a short abortive transcripts being produced. Alright, but now the polymerase has started, it is still stuck with GTFs and is blocked by this TF2B. And RNA polymerase is still linked with TBP. So it needs to escape the promoter, escape from these interactions with promoter-bound GTFs, specifically TF2B and TARA binding protein. And that is what happens in step 4, which is promoter clearance. So if we draw out this bubble conformation again for clarity, the thing that triggers the clearance is when these 3 to 7 bases transcripts grow in length to about 10 bases. In those cases, the RNA-DNA hybrid is forming a stronger bond, and such long transcripts are not easy to abort. So in this process of increasing the length of RNA, the B reader part of TF2B starts to get pushed back, and TF2B is eventually forced to dissociate away. Now because B reader had association with the DNA in the bubble, when it is kicked out, the actual available bubble length is now around 20 bases. But this 20 base pair bubble of unwound DNA is a bit too much for RNA polymerase. So this creates a thermodynamically unfavorable bubble, which collapses into a smaller bubble. And this collapse of a large bubble to a small bubble creates a force which causes a forward movement of the RNA polymerase. So now if we look at the structure of RNA polymerase with the smaller bubble, we have the RNA, which is still within the RNA exit channel. It hasn't come out of the channel because it is only about 10 to 15 nucleotides in length. But the RNA polymerase now has a stronger link with the RNA-DNA hybrid. This conformation is a bit more stable now. Now, simultaneous to the bubble collapse, TF2H also comes into the play. Here, especially the CDK7 subunit, which is the kinase part of TF2H, plays a crucial role in phosphorylating C-terminal domain of the RNA polymerase. As a quick reminder, kinases are enzymes that add phosphates to their substrates. Also, if you recall the introduction video, C-terminal domain is a disordered and repetitive sequence of 7 amino acids. In single letter code, these are YSP, TSPS. And these individual amino acids are denoted from 1 to 7. The Y is the starting position. So the kinase, the CDK7, acts on C-terminal domain. But this is only half of the picture. If you recall the full picture of pre-initiation complex, we've always had this enhancer complex contacting the promoter. So here, the mediator and kinases that are associated with enhancers also interact with C-terminal domain. The CDK7 from TF2H and enhancer-bound kinases add phosphate groups to serine 5 and serine 7 of each heptad repeat of the C-terminal domain. Just a technical side note, this complex of CDK7 and mediator that does this phosphorylation is collectively called CAK module, which stands for CDK7 associated kinase module. It essentially means that there are other kinases associated with CDK7. Now, functionally speaking, serine 7 phosphorylation, which occurs before serine 5 phosphorylation, is sort of a mystery. We don't quite understand exactly what the function of serine 7 phosphorylation is, but we do know that it is required for serine 5 phosphorylation to begin. And now, serine 5 phosphorylation is the important one, which is involved in the promoter clearance, because it breaks the interaction between RNA polymerase and the mediator complex on the enhancer. Also, serine 5 phosphorylation causes the RNA polymerase to leave the promoter and move forward, thereby escaping and clearing the promoter. As a result of this movement of RNA polymerase, it now synthesizes a few more nucleotides, and the length of RNA, as a result, increases to about 20 nucleotides on average. And if we draw out our visualization of RNA polymerase, it is now about 20 nucleotides away from the promoter region, with its serine 5 and serine 7 phosphorylated in the C-terminal region. And as we just said, the RNA length has increased, and now the RNA starts to stick out of the RNA exit channel, because it is a few nucleotides longer. But as soon as the 5' end of the RNA comes out of the polymerase, two special proteins bind to this 5' region. These two proteins are SPT4 and SPT5, which together are known as the DSIF complex. DSIF is a technical term for benzimidazole sensitivity-inducing factor. These two proteins, SPT4 and SPT5, are actually recruited onto serine 5 and serine 7 phosphorylated C-terminal domain. And it just so happens that this time correlates perfectly with the appearance of 5' end of the RNA from the exit channel. Normally, TF2F is sitting at this particular location, but when this promoter clearing happens, the DSIF complex takes the spot, essentially removing TF2F. Now, DSIF serves two very important functions. First is that it recruits capping enzymes, which modify the 5' end of the RNA, which improve the stability of the RNA. Second is that it recruits NELF, which stands for negative elongation factor. NELF, as the name implies, freezes the RNA polymerase. This freezing locks the RNA-DNA hybrid conformation in the RNA polymerase. So now it becomes difficult for the RNA polymerase to move forward. Essentially, it becomes paused. But this pausing gives time for the capping enzymes to do their job. So this pausing really has an important function in making the RNA stable via these 5' capping modification. And this pausing occurs somewhere around 20 to 40 bases away from the promoter region. And this pause is called a promoter proximal pause. And with this, we have covered the 3 steps that make up the initiation stage. Following this promoter proximal pause, the RNA polymerase has to overcome this frozen state so it can enter a productive elongation. And that will be the topic of the next video.